Good afternoon, everyone, esteemed guests, colleagues, and dear friends. We are thrilled and honored to invite you to join us in celebrating a remarkable milestone, the 25th anniversary of our CATS Lab, that is also known as Lab 25. We feel so lucky that this event also coincides with the 75th anniversary or Amrit Kal of our Indian independence. For a quarter of century, our lab has been at the forefront of computational research. It is synonymous with the algorithms, databases, and the structure. From unraveling the mystery of Tulsi genome to the secrets of cyclotides, our lab has continuously fostered a culture of innovation and collaboration. Our 25th anniversary celebration is an opportunity to reflect on our past accomplishments and embrace the future with renewed vigor. To commemorate this momentum occasion, we conducted a series of talks spanning 4 months from February of this year. In today's final session, we summarize and give a glimpse of those talks. We extend our heartfelt invitation to you, our esteemed guests, to be part of this extraordinary celebration. So we had 4 themes: sequence, structure, function, and medicinal plants in those talks. So without taking much of time, I would like to request Meenakshi to start the event with a summary of sequence. Thank you.
For the sequence theme, which is the theme in pink, we had 5 seminars, and the first, so I'll just share the highlights of the 5 seminars. So the first seminar was on computational sequence search approaches, and with special reference to motifs. So we discussed about structural motifs and how to identify conserved spatial structured motifs in sequences. And we also discussed about interacting spatial conserved motifs with special emphasis on the methyltransferase superfamily. Then we discussed about cascade sequence searches. So when we are interested in looking at a particular family members in different genomes, so we need to identify the related members and the distant members of the family. So during such cases, we used cascade sequence searches. So this is an algorithm developed in our lab. And so we also discussed about how to bridge the gap between the sequence space and the structure space. So we used remote homology detection methods and approaches such as the motifs, sequence conservation of motifs to identify members of the structure space in the sequence databases. And we populated all this information in the form of an update of a database called as Genomes. So in the second theme, we so all of us, we immersed ourselves deeply in the sea of protein domains. So we so when we bioinformaticians, we talk about protein sequences, we imagine protein sequences as a pearl necklace. So each pearl or each bead in it represents a domain. So a protein is basically the pearl necklace, and with consisting of multiple pearls, which are domains. So we and sometimes some of these pearls may be new to us. So we don't know what is the function of this domain and so on. So for such approaches, we have developed prediction of unassigned regions in for sequence analysis, where we help to identify the function and assign function to some regions whose function is not known in proteins. Then we also discussed about an approach called as ADAS. So some proteins, sometimes the pearl necklaces tend to be larger in length. So in such cases, it's very difficult to identify the sequence relationships if we take protein sequences alone and try to align them. So in such cases, we use an algorithm called as ADAS, which treats the proteins as this string of beads and then compares each bead with the other in the other sequences. So this the string of beads is compared across proteins, and then we identify the relationship amongst proteins. So this is very useful in cases where there are multiple domains in the protein. Then using all these approaches that we discussed so far, the sequence search approaches and the domain identification and analysis. So we present our lab studies on genome-wide surveys using two different families as examples. The first example is of tyrosine phosphatase. So we describe the work carried out in mining tyrosine phosphatases from the human and mammalian genome. And we also describe about the TIR domain homolog identification across mammalian genomes. So then we went to a very important topic, which is so all organisms, be it bacteria or plants or insects or humans, we like to sense our surroundings and then react accordingly. So in such cases, cues such as chemical cues and olfactory cues become very important. So we started the discussion by talking about our lab work on quorum molecule, which is involved in quorum sensing, which is the Luxes molecule, which help bacteria to sense the nearby environment. Then we discussed about GPCRs. We mined for the story on mining G protein coupled receptors in human and fish genomes. Then we also extended this seminar by talking about olfactory receptors. So olfactory receptors, be it human or insects, olfactory receptors are very important to sense the particular cues in the environment. And if we can, for insects, we can also target olfactory receptors in the form of insect repellents. Then we described an algorithm, InsectOR, which acts as an inspector and inspects protein sequences to identify odorant receptor genes. Then we discussed about another important class of proteins which are important in odorant detection, which are odorant binding proteins. So after this talk, we moved on to protein chewers, which are basically serine proteases or molecules which cleave proteins in the system. So we had a special emphasis on prolyl oligopeptidases also. So we started this session with a discussion on serine proteases mined from plant genomes, such as and two genomes, Arabidopsis and rice, for monocots and dicots. Then we also spoke about Drosophila serine protease, a class of Drosophila serine proteases. So this work was in collaboration with late Professor Veronica Rodriguez's lab. So from the lab we had they had this idea that there are some sequences serine proteases where the catalytic residues are not conserved. So in collaboration with that lab, we identified what would be the function of these such proteins, like which proteins do they interact with, and extended omics approaches for the characterization. The next, we discussed about a specific class of serine proteases, which is prolyl oligopeptidases. They are alpha-beta hydrolases. So we discussed about identifying these protein families using cascaded sequence search approaches from the bacterial genomes, and a specific application of these prolyl oligopeptidases, so they can be used for therapeutic applications such as in celiac diseases. Then, so we discussed about a very important problem, which is to look for nucleotide binders, that is DNA and RNA binding proteins. And this problem is very similar to looking for a needle in a haystack. So we, for the DNA binding protein part, we used several sequence and structural approaches to identify the DNA binding proteins, and we also described the work on the bacterial identification of plant and bacterial DNA binding proteins. Then we also discussed about the RNA binding proteins, where we had again used sequence and structural approaches to identify RNA binding proteins, and we emphasized on the fact that some of these these proteins can have moonlighting functions. They can interact with both DNA and RNA. I mean, the same protein can interact with two different type of nucleotides, that is DNA and RNA. Thank you.
Thank you, Meenakshi, for a beautiful summary of the sequencing. From sequence, now we move to the structure part or structure thing. Now I call upon Vikas to start the summary of the structure.
Thank you, Pankaj. So the next theme was about the structural structure of proteins, and again in this theme we had 4 different talks, and we started by talking about the protein-protein interactions. So the first topic in the first talk was about an algorithm called CUSP algorithm. So it basically, this algorithm tries to identify the conserved units in the structure of proteins. And what it tells is basically if you have a multiple sequence alignment, which was made using the structure-based alignment of the proteins, you can identify the blocks which are conserved. For example, if all the homologs of that protein has alpha helices in one place, or all beta strands, so those are called as the conserved blocks. And using this information, you can identify the deviant family of a superfamily members and so on. Next, we also came up with another algorithm called dock score. So in general, the protein-protein docking is very difficult, and most of the tools give several poses. So how do you identify which one is native-like pose or which one resembles most to the crystal structure pose? So to score those multiple poses, we came up with this algorithm called dock score. So it will tell you, given hundreds of poses, which one is more likely to be the native pose. So next, we also describe another algorithm called coil check. So it was developed to answer the question, like in case of coil protein, certain mutations of certain residues are deleterious in nature, while the others are not. So in coil proteins, especially, there are regions which are very highly stable regions, and there are weak regions also, which interact with other proteins. So to identify those regions, this algorithm was written, which was called coil check. And later, to build upon this or to make it a more general, another or the subsequent algorithm was called PP check, which basically tells the strength of the protein-protein interactions. And not just that, it also tells you about the hotspot residues present in the protein-protein interfaces. And it can also do computational alanine scanning of the entire protein structure. Moving on, the next talk in this theme was about the structural domains. So the first topic was about the algorithm which was written by Ma'am in our postdoctoral tenure called DIAL. So what it does is basically, so identifying the structural domain is not a trivial task. And to have an objective measure for that, this algorithm was written called DIAL. And later, this algorithm was applied to the entire protein structures present in the PDB to build a database called dial-derived database or DDBase. And using the same algorithm when applied to a larger database of structure, it was found that in some cases you have more than one chain coming together to build a single domain, which was pretty interesting. And therefore, it can be used to identify 3D domain swapping. So in domain swapping, you have like one domain of monomers get swapped with the other monomer of the same protein. And because of that, you have like domain swap interface or the non-swap interfaces. And this information was used to build upon several structural characterization of these two different interfaces, and which was used to further answer biologically relevant questions, especially in case of the diphtheria toxin. And next, once this algorithm was there, we also built a random forest-based classifier to identify whether a given sequence can have the potential to do domain swapping or not. And this algorithm was then applied to the entire genome. As an example, it was applied to the Tulsi genome and also to the human genome. Next talk in this theme was again related to protein domains, especially a class of subclass of the domains which were rich in the disulfide bonds. And it was called as the DSD base. So what it does is it basically tells you how many native disulfide bonds are there in the protein structure, along with how many you can engineer, which are stereochemically possible. And this code again was in all of these projects, ma'am herself had written some of the codes. And next, another structure-based sequence alignment database was developed in the lab, and it is kind of a hallmark database of our lab, which is being updated regularly. And it is called PAS2. So it tells you about the structure-based alignment of the sequence at the superfamily level. And it will also provide you the multiple hidden Markov models, which can be used to search in the sequence space to populate the superfamilies. And to further establish the related to the interaction in the domain, another algorithm was written, which is called HORI, or higher order residue interactions. So it calculates the direct interactions for each of the residues present in the protein structure. And further, like there are multiple modeling tools available, but whether the model structure is correct or not, to assess that, another algorithm called harmony was written. So it is possible that if you choose a bad template for your homology modeling, you can get a structure which is energetically favorable, like it will have overall negative potential energy, but still the structure will be incorrect. So it was shown as an example, like if you have immunoglobulin fold, you can model it as an all alpha helical protein, but that would be incorrect. To identify such incorrect models, this algorithm can be used. Next, we talked in the final talk was about the modeling approaches of different classes of proteins. One was the odorant receptor or the olfactory receptors. And in this case, again, the ORs from the mammals as well as from the insects were modeled. And again, it for the different species, the OR model structures were made available as a database. And we also talked about the modeling of the toll-like receptors and their domains, like the TIR domains. And lastly, we had another globular protein modeling, which is called CSRP3, cysteine serine-rich protein. And this protein was involving cardiomyopathies. And in this study, not just the modeling, but also it had served as a platform to do virtual saturation mutagenesis to give a structural rationale like why certain mutations are deleterious and to further predict which all other mutations can arise in the cardiomyopathy case, which can be deleterious for its function. Thank you.
Thank you, Vikas. Now we move from the structure theme to the function theme. Now I would like to invite Shaila to present the summary of the function.
Thank you, Mangesh. Hello, everyone. So as we heard about sequence and then structure, and then let's go to the implication part, which is the function. So again, in this theme, we had 4 talks, and each talk had a number of topics. So starting with the first talk of this theme, it was on protein 3-dimensional models are hidden jewels for function prediction. Here we had 5 stories. The first story was about R-struct FAM. So this was an approach to look for RNA binding proteins. So this did include sequence and structure-based approach, and using the HMM, how to find the RNA binding sites in the protein. The second story was on TGF, that is transforming growth factor, and it was an approach to look for the TGF binding protein. This question was initiated by Professor Vijay Raghavan, and here, the receptor, how it binds to the protein, it was well answered in as early as in 2000s. So Parantu, who did the project, was the first student of MAM. The third story was on YLHD, which is a fungal protein, and it is found in oil-contaminated areas. And this protein has a dual function, which performs both an epoxide as an epoxide hydrolases, also as an haloalkane hydrolase. So in this project, there was an attempt to look for the oligomerization state of this protein at different pH and how the oligomer plays different role as either of the functions. So the fourth story of this first talk was presented on deamylases and deacetylases from gram-positive and gram-negative bacteria. So it was found that in gram-negative bacteria, there were incidences of hexameric protein, but in case of gram-positive bacteria, there was a monomeric protein. And to surprise, there was another bacteria which was gram-negative, but there was a dimeric model. So to look for structural rationale of why another gram-negative bacteria, instead of having a hexameric complex, only has a dimeric complex. The modeling was performed, and then the in-house tool called as PP-check, which Vikas mentioned in the previous slide, was used to look at the energetics of the dimeric interface. Coming to the second talk of the function team was on when protein modeling go hand in hand with functional assays. So going to a step further, what happened when we looked for these models and try to correlate with the functional assay. So again, in this theme, there were 5 stories. The first story was on olfactory receptor. So this was by Dr. Shrishti from Shannon's lab. So in this story, they have tried to model the OR orchoreceptor of different insects, and then studied the ligand binding, which is coffee-pheronon, and with respect to the computational approach, try to see how good affinity it has. Also, the second story was on prediction of open state of thin cardiac filaments, proteins like tropomyosin and also F-actin. So here modeling approaches were taken further, and also some of the residues, key residues were identified, which on being mutated lead to diseases like cardiomyopathies. The third story from the second talk was on Hedgehog signaling protein, which is very important for tissue patterning. And the key of this project was that this was an ideal example of collaboration where the project with the prediction, the computational prediction, were very quickly taken to the wet lab experiments. The fourth story from the second talk was on PAS protein. So this is a metabolite channeling protein and how opening and closing varies. And this helps in the degradation of ring-containing compounds. And the fifth story in the second talk was on myotubulin, which is a mutant, which is a phosphatase binding protein. And then docking simulations were carried out, and also mutations were looked on in this protein. And in silico, these predictions were taken further to the experimental approach to see how the mutation affects the downstream signaling. The third talk of the function theme was on churning algorithms for enhanced function annotation. Again, in this topic, we had 4 stories. The first stories was presented by ma'am about CFG, which is clustering of functional groups, and the other is FASSM, which is functional annotation of sequence and structure motifs. So these 2 stories covers the approach using graph theory approach and also neural network approach, where they try to cluster the functional groups of a protein and how important it is for the functioning of the protein. And the second FASSM approach try to look at the motifs, the sequentially and structurally important motifs of the protein. The third story was on cascade search, where 2 homologous protein, how they are related to each other, was found through artificial sequences and connecting linkers. The fourth story was presented on CAPS protocol, which is widely used in our lab, where we mine for enzymes and protein of interest, look for the homologs, and then look for the approach to find the start point, and then screen for true hits to mine across different plants. So the fourth talk of this theme was on cellular assays, molecular dynamics, and ligand virtual screening towards understanding protein function. Again, this talk has 4 stories, which was presented in order of DDX24 being presented first, where we try to understand the role of this protein in case of planaria regeneration and what happened when this protein is knocked out. The second story was on ordinary receptor and an approach to mine for natural compounds, the small molecules, and on targeting one of the orchoreceptors from Aedes aegypti, how we can abrogate the downstream signaling. So 2 of the key findings were from the holy basil and the licorice, which can use the effective against the mosquito olfactory receptor. The third story was on prolyl oligopeptidases. So here we try to study using normal mode analysis, the ligand bound and ligand unbound form of the protein called as TOP. And the fourth story was again a virtual screening approach for the against the SARS-CoV-2 virus targeting the NSP1 protein. This was done during the COVID times and was completed within a month. Thank you.
Thank you, Shweta, for summing of the function theme. Now we move from the function theme to the medicinal plant theme, and I would like to invite Shafi for this.
Thank you, Pankaj. Okay, so in this series, this is the last thing, and we move to medicinal plants and collaborations. Okay, so and in this last theme, we started with plant stress management, and where we had 3 sessions altogether. And the first session, Dr. Shamir Kader talked about stiff and in-house algorithm in the lab, where we predict cis elements in the promoters of the gene. So currently we have an algorithm. Also, we have a database. In the database, we have model species like Arabidopsis thaliana and Oryza sativa species. And next, we talked about a collaborative work from Professor Atle Bond's lab from Norway. Dr. Pankaj Bara talked about combined and single stress in Arabidopsis. He collected several ecotypes from worldwide, and he looked at the promoters and he applied the stiff algorithm and analyzed them. And in the third session, this is also again a collaborative work with MSRF, Dr. Gayatri Venkataraman. And we looked at sodium transporter in rice species, and we looked at salinity stress and how structure variation in this transporter affect the salinity tolerance in the cultivated and wild variety of rice. Okay, so moving to the second talk in this theme, we taking through to medicinal world of medicinal plants. And we have selected few well-known medicinal plants. First, the sequence Tulsi, led by Atul Upadhyay, and this was a whole teamwork from the lab. Everybody from the lab participated in this, and we sequenced the genome and transcriptome of Tulsi, and we compared both Krishna and Rama Tulsi for the transcriptome. And following to that, we selected another plant, fenugreek or methi. And for this, we have done transcriptome sequencing, and we looked at different tissues of this plant and selected few important metabolites in this plant, like trigonelin, diosgenin, 4-HAL, and which are important for antidiabetic properties. And we have quantified them and we have analyzed them in different tissues. And in the third session in this talk, we talked about Perendi, Cicer quadrangularis. And this plant is known for bone healing and other important biological activities. We have done genome sequencing and we have done a genome announcement also for this. Okay, so moving to the second part of this world of medicinal plants, and we studied drumstick tree, Moringa olifera, where we have done transcriptome sequencing and we selected 5 different tissues. And we looked at several secondary metabolites involved in various biological activities, and we quantified them, and we also looked at the minerals in these plants, and there are the abundance of all these things we have reported. And following to that, we collaborated with TDU and we looked at another wild variety of this drumstick tea, and we we compared with the normal drumstick tree and how it is different in terms of antidiabetic property, and we have studied them. And in the third session, we talked about Shangapushpi transcriptome. This is a collaboration with Dr. Radhika Venkatesan and also Professor Balram. And Neha Kalmander, she studied the cyclotides, and from the transcriptome, she selected the cyclotides and studied Alzheimer's and other activities in this plant. And coming to the fourth talk, we not only talked about plants, we have talked about bacterial pathogens and viruses. So here, the first session, it's a collaborative work with Professor Umar Ramakrishnan, where we studied one of the invasive plant, Landana. And here, Praveen Prakash talked about the population genetics of this Landana species throughout, and then Advait Joshi talked about the genome of this Landana, what we have revealed. Then in the third session, Meenakshi Iyer, and she went to Kenya and she had sequenced the bacterial pathogen Neisseria gonorrhea, and she talked about antimicrobial resistance in that. And the fourth talk, Abhishek Sharma, he's a joint student from Professor Sudhir Krishna, and he has looked at dengue viral sequences and he has analyzed the mutation structural analysis in them. Yeah, so that's all. Okay.
Thank you, Shafi, for summarizing the medicinal plant theme. Now we move on to showcase our history of our clusters. For this, I would like to invite Advait.
Thank you, Bhavesh. Good afternoon, all. Thank you for coming for the talk. So before we proceed to the computing history, yeah, okay, should be on the video. Okay, before we proceed, I just ask all the participants who have joined as panelists. Hello to all CAPS lab members. Please, can you turn on the video? We'll take a snapshot. Thank you. And also, if you have any points, questions, comments to talk about, do raise your hand. We'll take it up after I finish my presentation. Great. Okay. So if you have any points, do raise your hand and we'll take it up after I finish my presentation. All right. So we heard about a lot of topics today, going from sequence theme, structure theme, function theme, and medicinal plants. And in many of them, you heard the word evolution. But what I'm going to tell about is the evolution of computing at CAPS. Okay, this is very close to heart because everything depends on how we deal with these computers, and these features an important factor which are systems and the system admins as well. So if we say about the cluster evolution. It started way back in early 2000s when we got our first cluster, which was called as Linome, which had 20 nodes. And subsequently, we got a couple of clusters, called them Quadrum 1 and 2, which together built up to 30 nodes. And both of them were funded through Wellcome Trust Grant. Later on, we got a monster powerhouse computing cluster in 2010, which was funded by Department of Biotechnology, Center for Excellence Grant. And this was a really workhorse with 702 cores, and we relied heavily on this. I'll come about that and talk about that in subsequent slides. And to follow it up, next year, 2011, we got our another virtual SMP machine, which had 96 cores, thanks to the HFSP grant. And finally, at a latest entrant to our cluster compilation is Panchashata machine, which has 512 cores, which was through our NCBS plant core grant. We have a couple of GPU machines also, GPU 1, Pascal GPU, which have 4 and 6 GPU cards according, respectively, funded by HFSP and NCBS core grant. So how do we have this expansion in computing power? So if you see, okay, this is the in 2000, this is the 1,000 rupee note. Now it's irrelevant. Now the 2,000 rupee note will also become irrelevant in few months. So we got our first cluster Linome in 2004, and in this was just a 20 node cluster, whereas in 2011 it went to 96 cores, this virtual SMP. It was like a first 100 node cluster. And in 2017, it increased to 500 cores. So these expansion in computing power is required and is necessary, as well as there is also a requirement, ever increasing requirement for storage space. I'll come to that in subsequent slides. So one of the powers that I was talking about, the SGI machine, the UV1000 SMP cluster, also was captured in news. And you can see over here that it is placed in the basement of IT, which requires heavy cooling. So how do we get these things to the IT department? So this is how we meet our clusters, use heavy cranes and all that, and bring them to the IT section. So this was a fun memory to bring the machines to their home and then work on them. So along with clusters, there are small players as well, which are desktops, which you see in our lab. So we have some interesting ways to name these desktops, either based on what project we are working on, or some special ones which are like serial relies. These are the softwares what we have, and dedicated machines for those softwares. So these IPs are the boring names. Again, these are older IPs. We have a newer set now. Okay, so what goes into getting a cluster? So this is an important part, because we have to judge what are our requirements, which kind of jobs we want to run. And again, money also matters. So how much is the budget? Accordingly, then we have to go for either GPU, CPU based or storage intensive, memory intensive kind of a machine, or catering to a particular software like Civil or Schrodinger suite. And after we get quotations, we have to select based on that, and we also have to extensively do the benchmarking, because we have heavily invested in that. And then it can cater to, say, genome assembly, MD simulations. So such programs are used to benchmark it, and finally the cluster is business. All right. So as I was telling, this features systems and system admin as well. This is an important part. You need a root permission. You have to run to any of these guys, and then get your programs installed. So these are some of the CAPS members who have worked as system admins. A couple of them are pseudo system admins as well. We stole the root password and are using it. Okay. So we do another important thing. When we have such a huge computing power, it has to be managed properly. So one of our system admin, Dr. Roman Matthew, he developed a portal called as Capsters. So in this, we get like this is the dashboard, and you can see we can request we can request new jobs what we want to run, start date, end date, how much, how intensive it will be, and then we also get a list of servers clusters that we have at our disposal, and finally a scheduler in which we can see what all jobs are running, and then accordingly system admins approve the job or they deny the job. Denial doesn't come that often, but yeah, system admins have a tough time doing that. So that's how we manage our clusters. And yeah, so since the IPL ended just a few days back, I thought of putting this slide as a CAPS Premier League, where we don't pit these machines against each other, but we do have to struggle around to see which machine to use, and each of them have their own plus points. And in the archives, then you can see particularly it goes with floppy disks. We do have some floppy disks as well, which are very old ones, but LTO tests, some of you may not know, but those are very robust one, and couple of older programs are over there. But now we have to rely upon system admin discussion as well as our own judgment how the job in how intensive computing is required for a particular job or how much storage, and then we go on with playing around with these machines. All right. So I'll just ask any of the CAPS alumni who want to speak up.
Hello?
Yeah, you're audible. Yes.
Yeah, okay. Well, thanks everyone. And thank you, ma'am, for giving me an opportunity to present the work which we did together. I'm very pleased for that. And it's always been a very proud thing to be associated to CAPS, and I celebrate it every day. I think I can just say that the lab was just the right place for getting the right foundation, the right mentoring, and the right direction for all my I can completely rely on building my career on that, and I think there is lots to learn from the lab apart from the great science which we do. I think personally I have learned a lot from the lab, on the personal front as well, and professionally, of course, how to deal with collaborators, how to manage collaborations, how to do science, of course. And yeah, and I'm really, as I said, I'm really pleased to be part of CAPS and will always be proud to be part of CAPS. And thank you for giving me this opportunity. Thank you.
Thank you, Soni. We'll now hear from Rajas. Hello, Rajas.
Hi, this is Rajas. Just to give a brief introduction about myself, I'm an assistant professor at the Yenagoa University. So my journey with CAPS began in 2015, where I was a final year MSc student looking for internship project. To be frank, I was an outsider to the world of bioinformatics, and I approached ma'am, and she was glad. She was really kind to accept me and into the CAPS group, and that's where my journey into the world of bioinformatics began. And I couldn't have been a happier person since then to have been part of the CAPS group, to have experienced the world of bioinformatics, and how to have learned from Professor Saudamini lots of things, including both science as well as things other than science. And I'm forever thankful for that.
Thank you, Rajas. Thank you, Rajas and Soni, for your nice thoughts. So we heard from last week when we had our last theme from the function themes last talk. Okay, we heard from Jitu as well as Uma some nice words of CAPS. Today we have one more recording.
Good afternoon. Understanding biological phenomena in the natural world is a fascinating problem. Yet these are very complex phenomena that require a great deal of effort to unravel the complex mechanisms that underlie the beauty of what we see around us. Historically, there's been a great deal of emphasis in using experimental approaches in understanding biology. But as we go along, the value of using predictive approaches to accelerate discovery in biology has become very evident. Key among these is bioinformatics, the ability to look at the DNA sequence of an organism and predict the composition of proteins within these cells and how that might contribute Likewise, the ability to look at the sequence of a protein and predict its function is surely one of the most influential areas of modern biology. The CAPS lab at NCBS, led by Professor Saudami, has been engaged in this work for 25 years. And on this occasion, I would like to congratulate Professor Saudami and all former and current members of our lab on their exceptional contributions in this area at NCBS. We also look forward to many more exciting discoveries from their work as we go along, trying to unravel the beauty of nature in its details. Thank you.
That was nice. Thanks to hear from our colleagues here at NCBS. So just before moving to the next part, we'll hear from Mahantesh. And where's Bhavika? Mahantesh, can you come on there?
Yeah.
Hi, welcome.
I'm audible.
Yes.
Okay. So good afternoon to all. I am associated with the CAPS lab from 2009, and I'm very much grateful to Professor Saudami ma'am. Since my academic background is agricultural sciences, and ma'am is so kind to give me an opportunity to join the lab as a collaborative PhD student. And since then, my journey of learning with respect to the bioinformatics began from the zero knowledge, and got a lot of information from ma'am, and gave me an excellent opportunity to collaborate with US, Bangalore, as well as with some international collaborators. That was the time we got a lot of exposure and a lot of learning experience. And as a student, we are very much thankful to ma'am for ma'am's dedication, commitment, sincerity. We have not only learned the science from CAPS, but also we learned a lot of the things from ma'am. Seeing ma'am's dedication towards science, and again giving excellent guidance to the students, and that what we learned from the CAPS lab today, we are giving our best to the scientific world. And what I would like to say is thank you so very much to you, ma'am, and once again congratulations for contributing enormously to the scientific world. And once again, thank you, ma'am, and congratulations as well. Thank you.
Thank you, Mahantesh. So in interest of time, we'll just move to the next part. Bhavika will catch up with you in some time. Okay. So over to you, Pankaj.
Thank you, Andre, for showcasing our clusters. In the last, I would like to invite our mentor, Professor Asha Dhamni, to say a few words in the concluding remarks. Over to you.
That was a nice time spent, isn't it? To get a summary of all the talks we've had for the past 4 months. First of all, I would like to thank the audience here, as well as the virtual audience we have had for the past few weeks, who have been very patiently listening to our stories. And we have learned a lot from the feedback we have got, the questions we have had, and so on. And secondly, I would like to call up on the CAPS core team, as we call it. There are 5 of them who had presented just before me. I would request them to just get up a little bit so that everyone can see who you are. And without them, I think none of these virtual sessions would have been possible. And a big clap to all of them. Okay, so there we are. So that's what we call as a core team. Plus, we got a lot and lots of advice from Sonal, who has been very enthusiastically helping us from day one until today. And back end, I know that all this was possible because of the outreach committee that consists of Uma and Mukund, and thanks to them as well. Plus the instrumentation team, who have been completely supportive of our requirements, and put up with all our kind of tantrums. Okay. I wish to just remind ourselves that those of you who have listened to the online talks before would remember that there have been a consolidation at the end of every theme. And that means if we had 4 talks, they get consolidated at the end of the theme, which we tried graphically. And this is how it goes. The story whichever story we have projected will come to the left, and then the people involved. And then the blocks represent how much time they had spent in the lab. And these are like 2-3 people, and it's a very long project. It started sometime in 2003 and it's still ongoing. So this is on the plant stress management. And then what we have tried to show is in these little symbols are the publications. So this kind of provides us a realistic picture about how much time each of the projects have taken, how much time the people have been involved, and at what point do we get the publications, right? So I hope this will kind of bust some of the myths that have been floating around about bioinformatics work. And I speak for every bioinformatician in the whole world, which is to say that none of these are rapid, easy, or any of such things, because for each of them, we spend at least 3 years on the project and 1 year to get it published. And we have had always students who stayed at least 5 years. Of course, that's something I wanted to mention right in the beginning. And then I'll just switch on to something quite lighthearted for a little while, if I may. Okay. Back in 1998, when I was absorbed, I think I was the 12th faculty to be absorbed. And I must first of all congratulate 2 people, or maybe 4 or 5 people, who had the courage to absorb me in NCBS. Because, and these include people like Professor Vijay Raghavan, the director then, Professor Jayant Udgonkar, the dean then, and then I'm sure many more people were involved like Matthew and Satyajit Mayer. So they have all played a role, and I say they had the courage because at that time or even now, I don't have any nature papers, I don't have a science paper, I don't have one in molecular cell or any of these. And I know some of the faculty who are being absorbed or even being considered have multiple such papers, and we are still thinking, should we take them or not? So that is the kind of trend we have, which is very good for the center. But looking back, I could have easily been one of those very unproductive people. So this is really just because of your vision and the risk they took to absorb me. And then other features is not only I was the 12th faculty or whatever, I was also the first mother scientist, mother PI. And that meant literally, since I had just come from the UK, I said, we will move to the new campus, and many people were trying to buy me that idea. And I remember Jayant told, we get milk, we get newspaper, why don't you move into the new campus? And I said, no, until and unless we have a creche, I'm not coming anywhere close to the new campus. And that's where it started, because we have wonderful support, because every one of the faculty donated their children, their babies or children, whoever, what best we can ask for, into the creche. And it began in that small way. Of course, money was also pumped in. We didn't have any regrets, because we can see that it's one of the thriving facilities in the campus, and I'm so happy it stays on. All right. So that was not an issue, because everyone supported it, and also we could just move on because of the support we had, not only from the creche point of view, but also in terms of the academic support, because almost everyone encouraged me to just proceed with the work, and it was just like any other PI. I was trying and struggling in the beginning, of course, but what was strange and so perhaps unique for me as the 12th PI who joined NCBS is that okay, when I joined, I was very content with myself, very pleased with myself to have first of all got this position, because I had rigorous training and stereochemistry from my PhD days in Indian Institute of Science. And then I had been in the UK, in the heart of where bioinformatics grows very well. And I had been very appreciative of protein folds. And I had lots of interest on programming, because it used to be our primary preoccupation. So I thought I have everything I need, and I was very interested in biology. So I thought everything is going well. But what happened was I was perhaps the only one who was doing hardcore computing, right? So wet lab here, wet lab there, wet lab there. And some people are talking about Drosophila, and then someone is telling about stress, mice under stress. And these words are reeling all around me, and I know that I'm a minority. So it did take a long time to accept myself as a minority and for the others to accept me. Okay, so that seemed to be the main issue. However, I am truly very, very thankful for that situation, because that really made me to improve and also gain more potential, kind of present and get the best out of ourselves. And this meant we continue to work hard. So this is how it is reflected, I suppose, in terms of the number of publications. But that's not all, because like self-motivated manner, we also went on to get many grants. These were internal grants which come at every 5-year period. But we also used to try, and many of them get turned down also. But the external grants that we wrote and did work out are mentioned here over time. And also some of the other, not only Department of Biotechnology, but some of the competitive sort of grants are also marked up here. Plus the recent ones, I could say, started with the Wellcome Trust, and then the Human Frontier Science Program grant, and then the GIC Post Fellowship and so on. But recently we have had a few more, which is the CFIPRA. Okay, so this helped us to be more self-motivated and try out, because we do get funding internally, but that doesn't stop us from trying more. And focused projects always help us to be more collaborative. So that's what we learned. But as a new PI, I was also going through the same anxiety and struggle, because whereas we get training on doing the academics, we don't get trained on financial management, a little bit on people management, because we do mentor as postdocs, right? So I'm sure people around me were a little concerned that, because I was quite shy and sensitive, and so it seemed at least then. But I realized that whereas there is a concern to attract and retain students, what is more important is to be ourselves. And that's what I began doing. To be ourselves, however you are at home, you do likewise behave, treat people well. And of course, you're dealing with mature students in the lab. But that definitely helped, because a lab could get more and more students. And this is unfortunately the year of 2009 and 2010. We had the opposite issue, because this was the year when we had lots of national fellowships, and people, especially students from Pune University who had substantial programming training, chose our lab. And we could call this the Cambrian evolution, as it were, because there were a number of students pouring in. And this means we were able to escalate our kind of opportunities was there, and escalate our questions to kind of fine tune on programming intense sort of projects. It was possible, and really it helped us to ask bigger questions. But it also meant at some point of mine, at least 2 times, I had a situation where 4 students were writing theses at the same time. But then being computing people and being so objective, I used to mark when they submitted their first version, second version, and according to the date, as fair-minded as possible, I used to give it back to them. So this is all getting recorded, and I would say really transparency is the main thing, because you can then make sure that you can deal with a situation such as multiple students writing theses and so on. And this is a picture, 1 slide each. This is what we had asked from the old students. Then this was when we were kind of realizing our 20 years. And this is now one of the students, Raghu Prasad Rao, as a student, how he was and how he is these days. Yeah, so he's a faculty somewhere. So this, I'm not going to show you all the slides, but just to say that objectivity and transparency is important for us. And this is the electronic diary called Dot Project, in which I have been recording, I think, for the past at least 10-15 years. And it helps us to know what projects are pending, when we are going to discuss, with whom. And also this can be subdivided in fact, my daily schedule can be broken into every 15 minutes. And we keep track, and it's transparent. It's transparent in the sense whoever is in the group, namely the lab members, can look at it, so they know that their turn can come and they can ask when they want to meet me. They can always solicit that. So there's a lot of advantage. I thought I will give you an idea about how things were going. But if you're handling many students and so on, you might have caught us looking very preoccupied, very serious, deadline driven. So this is because we were having many students and projects. And later I'll come back to the question about how do we manage projects, because it was the first question that Raghu asked me after he joined NCBS formally as a faculty. But to just tell you that we're not serious all the time, because we go for retreats, and every time a student submit thesis, there is a treat, and then they defend the thesis, there is a treat, etc. Plus, we also kind of amuse ourselves by means of creating these sort of designing some t-shirts, and my students have always been coming up with some very exciting ideas. So these all go into the fun part, because that's just to make sure our sanity levels are high, and we have many other ways of making sure it is high indeed. Okay, so now coming back to the serious parts. Okay, we have heard from the CAPS core team members a number of projects, and we try to put them together, weave it around a theme and so on. But I realized that I still have not covered some of the projects, especially even though they were published, somehow I just could not cover it. So to be fair, I'll just rapidly put them across. I may not even talk about it. However, just to say that this was the very first formal collaborative project which we had after joining after I joined NCBS, which was to look at the single nucleotide, single residue mutations in nucleosomes, and this was with Shiva Shankar. And then this was the second formal collaboration we had with Apurva Sering, which was to model and do dynamics. And this was in the year 2007. And the reviewer said, would you step it up to 30 nanoseconds? And oh, what? 30 nanoseconds? It looked like a very big number then. But now people do in microseconds easily. So that was how the scaling was going on. And then these are some other projects that I had not had, we did not get a chance to present, but there they are. So these are all kind of different projects where we could participate. And the next slide is going to be specifically on plant projects. And we do have a server which kind of helps us to inquire and find out if we have a gene of interest, which tissue is it likely to be more expressed and so on. And then we had recently collaborated with 2 labs in BIRC, Atomic Research Center, one on the GroundNet project and another on radioactive resistance that has been going on. Some people in Kerala coast have been naturally resistant to the radioactive radiation. So the question was, how do they manage to remain healthy? So some transcriptome studies were done. These were all about different projects that got left out as far as I could remember. And I'm unfortunately only had covered the published projects. So the ongoing ones, the unpublished ones, we have not talked about. All right. And then the question from Raghu had been, how are we managing so many projects? Yeah. So I asked myself this question, and then what we did was some kind of informatics. So we the current students and also the previous alumni, we just grouped them into categories, and those who have been handling 1 project, 2 projects, as much as 10 projects or more than that. So then you can see the histogram shows that many of them have indeed been handling 1 or 2 projects, which means it's fine. But there are also people who have been handling more than 10 or more projects. So that actually it seems to be the main reason why we are able to, produce more publications, because publications are as hard as any other area in biology, because we do go through a lot of reviewers' comments, and it does take time even to complete the work or whatever. But also because it's very less demanding on money, because we can just start off a project with nearly no money involved. So that is another incentive. And this video, which we have made, is kind of a network picture. What we can see in this squares are people, and what we have in this blue color are the projects. This is a very real picture where you can begin to appreciate that I'm somewhere here toggling around. And then that is Meenakshi. So for instance, that's an example to show that multiple projects are being handled by 1 person. And now she will go back. And then you can also do something which is protein centric. You can focus on one of these blue boxes, And for instance, Tulsi project, we have heard a lot about it because everyone in the lab at that time had participated. So you can see we focus on Tulsi, like many connections. And then, yeah, so that's how our story goes. And I would like to say, from the bottom of my heart, I cannot thank all my collaborators. Go and tell them, hey, thank you, hey, thank you. I cannot say this, but at this juncture, I would like to thank every one of them because a lot of the work became possible just because of this sort of thriving environment and the people who are willing to collaborate with us. And what I've tried to do is to categorize them depending on those themes that we have heard, sequence, structure, function, theme. So you might find that the same person is more than one theme, unfortunate for them, but yeah, they are there. But I must say, in terms of the publications, I explained to you the environment was your kind of a minority, and most of the others are doing wet lab. And I must say, however, there was enormous support and positive encouragement from them. It's not like it's not as though they would not encourage us. Plus, I had enormous support from my husband, late Professor Srinivasan, who kept encouraging me to keep going. So these are the collaborators. And finally, the rest of the lab members, you will find that they are all put on their CAPS t-shirts here, so you will find them. Plus, there are a few more whom we could thank. But before that, let me just recall what Jitu said, because in his presentation, which was put up last week, he said, you all have been doing well, you have done 25 years and wish you 25 years more. And that was a very sweet message. But what I would like to say to him is that we have learned so much, but we will like to learn a lot more. If we really do stay on those many years, I hope to become a better person and also a better scientist in the process. Thank you. This is just kind of a scroll to tell people involved, and the collaborators come again, but then the more people, there are IT people, there are administration people. So yeah, thanking all of them.
In the audience, anyone has to say anything or few words? I don't know if you remember. I first visited NCBS in 2000. I was in grad school, and I didn't even know about the place. I happened to come, and there was a talk happening in this auditorium. And I've been trying to think who gave that talk because I didn't know anybody here. And I realized at some point it was you. It's a picture of protein, and I didn't even so outside of biology. I didn't know that people could have such knowledge of these molecules. And we chatted outside there.
Mm-hmm.
So you were, I think, literally the first person in NCBS.
Yeah. Oh, nice. And you're staying on. Thank you, everyone. Oh, there is tea outside. Please join.
I wanted to thank you.
Thank you, ma'am.
In the 1.5 years that I've been here, I had one of the best times working with your lab as a group. Everyone was involved, really good discussions of even tiny things, why we should treat like this, why the color theory should be like this. I could have deeper discussions. They gave me that much time. They were very flexible, very understanding. I learned a lot from them. Some comments went from mine. So I enjoyed a lot working with you. Thank you.
Same here.
We are ending this. Thank you everyone for coming.